so if anybody is not able to hear me, please put it in the chat. We are supposed to have started the class at 9 o'clock. It's already 9 o'clock. So we will go ahead. Those who have not logged in, is up to them. It's already 9:02. We are dealing with the both reproductive, endocrine, and urinary system. And endocrine and urinary reproductive system are very closely associated together because the endocrine system regulates the reproductive functions. And that's why they have been put together. And then apart from that, the reproductive system and urinary system also have a common origin from the intermediate mesoderm. That's why they are also put together because of that common origin. And some of the disorders of the reproductive system can also impact on urinary system. That's why these systems have been put together. So we are going to start with the endocrine system. We have already done the endocrine system when we were looking at various types of glands. And we classified glands as those which have ducts and those which do not have ducts. So endocrine are those which do not have ducts, and the hormones are transported by blood, by lymphatics, by CSF, cerebrospinal fluid. So that's how hormones can be transported, not in ducts. So the actual glands secrete the hormone by process of exocytosis to the perivascular space, that is within the interstitial fluid. And then from this interstitial fluid, the hormones can then pass into blood or the lymphatics. So we are going to look at these organs, their locations, their structure, and also mention about the hormones they produce and some of the hormones, functions of those hormones, and the disorders that sometimes do occur because of either excess production of the hormone or less hormone being produced. So that is how I would like us to deal with this. So for endocrine system, we let me just get a pointer so that we're able to follow each other. So for endocrine system, we note that we have several glands of the endocrine system. Sometimes we do classify them into central and peripheral endocrine system. So central and peripheral. Sorry.
Excuse me, sir. I think you're muted.
Okay, sorry for that. Let me just repeat. We have the central, what I put here in this is central endocrine organs, and these ones are peripheral endocrine organs. And I've said central endocrine organs, some of them regulate peripheral endocrine organs. Among the central is hypothalamus, which is part of the brain, just below the thalamus. It originates from the diencephalon. We have the pineal gland also originating from the diencephalon, and the pineal gland is in the roof of the third ventricle. We have the hypophysis occupying the hypophyseal fossa in the sphenoid bone. That is the hypophysis. That is also connected to the brain, to the hypothalamus. Then the peripheral, we have the thyroid gland in the neck region, around the thyroid cartilage, thyroid and cricoid cartilage. We have the parathyroids, which are just posterior to the thyroid, and enclosed within the capsule of the thyroid. Then we have the thymus. If you remember, we have done the thymus before when we were looking at the immune system. The thymus is in the anterior mediastinum within the thorax. Then we have suprarenal glands above the kidneys, the suprarenal above the kidneys. Each of the kidneys will have a suprarenal gland. And then the islets of Langerhans, that is in the pancreas. So the islets are part of the pancreas, but they are the endocrine part of the pancreas. And they are islets, they are islands. They look like islands within the exocrine pancreas. We'll be looking at the details. And then we have the gonads, both the testes and the ovary. Apart from this, we also have what I consider as diffuse neuroendocrine system. Diffuse neuroendocrine system comprises of either a cluster of cells or individual cells which secrete hormones. And these are in various parts. They are in the respiratory system, the respiratory tract. They are also in the digestive tract, quite a lot of them in the digestive tract. We have them in the heart. We have them in the kidney. So these ones are cells which secrete hormones. They secrete hormones, but are either as individual cells or groups of cells. Where there are groups of cells, we can call them neuroendocrine bodies. The neuroendocrine bodies, we use the word neuro because these ones originate from the neurocrest cells. So these are the organs we are going to look at in terms of their structure. Now, we know that hormones, usually we have regulatory mechanisms, and hormones are produced by the glands. They're transported by the blood. Some of the hormones from the central endocrine system, that is from the hypothalamus and the hypophysis, are transported to the blood to the target organs. Those target organs now produce hormones. These hormones then can have a feedback mechanisms so that you don't have excess production of the hormones. Occasionally, you might have excess production of hormones when you have tumors or hyperplasia of the organs, overgrowth of the organ. Then in that case, you will have excess production. But the normal mechanisms are such that the hormones that have been produced influence those particular cells which produce the hormones. These we refer to as feedback mechanisms. So we have the direct feedback mechanisms and indirect feedback mechanisms. In the direct feedback mechanism, we have either long loop or short loop. Let's take a case of a hormone being produced by the hypothalamus So the hormone being produced by the hypothalamus will influence the hypophysis. The hypophysis produces a hormone. That hormone then goes to negatively influence those cells that produce the hormone from the hypothalamus. So they have negative feedback mechanisms. We have cells within the hypothalamus and hormones produced by the hypothalamus that are inhibitory. So this is a short loop. And then for the long loop, where we have the target organs, let's say the testis or the ovary, produces the hormones, and those hormones can influence the hypothalamus, which is supposed to be stimulating the production of those hormones by the hypophysis. So this is an indirect long loop, whereas the direct short loop, a long loop, the direct short long loop is where we have those hormones from the target organs, like the ovary or the testis, directly influencing the hypophysis so that those hormones stop being produced. For example, you can have follicle-stimulating hormone being produced by the hypophysis. It stimulates the development of the follicles within the ovary, and those follicles then produce a hormone, estrogen. The estrogen then negatively inhibit production of further follicle-stimulating hormone so that we don't have excess production of the hormone. Or the estrogen will inhibit the releasing hormones, gonadotropin releasing hormone, which is supposed to stimulate those cells that produce follicle-stimulating hormone. That is now in the indirect long loop. And then the ultra short loop is where we have the hormones negatively influencing directly from the median eminence, as we'll be able to see. So these are negative feedback mechanisms. We also have positive feedback mechanism. In the positive feedback mechanism, a good example is oxytocin. Normally, oxytocin influences the milk let down and also contraction of the uterus. So during lactation, when the baby is suckling, then neural stimulation of the nipple gives a feedback to the hypothalamus, and then the hypothalamus produces more oxytocin so that you can have more milk being produced. Similarly, when the baby is at the cervix, the head is at the cervix during delivery, the pressure applied to the cervix, nerve endings from that particular part will give a feedback mechanism so that you have more oxytocin being produced so that the uterine wall can contract to push the baby out. Those are positive feedback mechanisms. The rest are negative feedback mechanisms. Let's now look at the structure of the general endocrine organs. It's an organ. So like all the other organs, they are normally solid organs, and they have a lot of connective tissue. The first connective tissue is what covers the organ, that we refer to as a capsule. That's a fibrous connective tissue, dense fibrous connective tissue. This is to protect the organ. That dense fibrous connective tissue penetrates into the organ either as a septa, if it's a very thin penetration, or trabecular. And this may divide the organ into lobes and lobules. We'll be seeing that case in the case of the thyroid gland, a very clear case. Also, the connective tissue, which is now the stroma, supports the nerves, the blood vessels, as they enter into the organ. And the area where these structures enter into the organ, the neurovascular bundles, is what we call the hilum, or the hilus of the organ. And then we have the parenchyma. The parenchyma are the cells. Those cells secreting the hormone are usually arranged in either clusters, a cluster of cells, or clumps of cells, or sometimes in codes. Can be 2 layers of cells forming a cord, or 3 layers of cells forming a cord, as long as each of those cells is close to a blood vessel. They might also form some nest-like structures, or they might be anastomosing cords. So these cords join each other to form a network. That's what they're calling anastomosing vessels. So we have a very well vascularized network of blood vessels, and this comprises of sinusoidal capillaries or fenestrated capillaries. Because the molecules that are being released, the hormones that are being released, are very large, they cannot pass through a continuous capillary. So those which are smaller will pass through fenestrated capillaries because they are window-like apertures that allow the passage of the hormone into the blood vessels for onward transport to the target organs. So this is the general structure that we will not be talking about. Again, each of those will have a capsule, it will have a septa, it will have a trabecula, it will have the parenchyma, the cells, and the vascular network. Apart from this, of course, we have the ground matrix in which the cells and the fibers are suspended. Now, as we look at these cells, as we look at the hormones, we need to understand what is the biochemical nature of these hormones. Because we are going to describe the cells that produce these hormones. We are going to look at those organelles that produce the hormones. So we must understand what is the chemical nature of the hormones. Hormones are of various types. They can be peptides, and these are amino acid derivatives from proteins. Among the peptides, we have the adrenocorticotropic hormone released from the pituitary gland, oxytocin, antidiuretic hormone, also called vasopressin, released from the neurohypophysis, but actually produced by the hypothalamus. We have the somatosensoric pancreas, the glucagon hormone, the insulin, all these are produced by the pancreas. We have calcitonin produced by the C cells of the thyroid gland. We have the parathyroid hormone, and then produced by the parathyroid gland. And then we have atrial natriuretic peptide, also called atrial natriuretic factor, which is produced by the cells of the heart. So these are peptide nature. So we know what peptides are. We are going to eventually look at those organs, and that as we describe them, we are going to look at the organelles that synthesize those peptides. The others are proteins, protein in nature. The growth hormone, which is very important in growth of muscles, growth of bones. So the growth hormone is a protein in nature. And then prolactin. Prolactin, also, all these two are produced by the hypophysis. Growth hormone and prolactin are produced by the hypophysis. Prolactin, that is very important in the development of the mammary gland. The other ones are glycoproteins. Remember, this is a glycoprotein, which is having carbohydrates, oligosaccharides, and proteins combined. carbohydrates and proteins combined. And usually these are combined by the Golgi apparatus. So we'll have very well developed Golgi apparatus in these particular organs that have, or the cells that secrete these particular hormones. The thyroid stimulating hormone, which is from the thyroid gland, the luteinizing hormone, and follicle stimulating hormone. These ones are from the hypophysis. Erythropoietin, which is produced by the kidney, cells of the kidney. So these are glycoproteins. Being glycoproteins, you can stain for them used to demonstrate carbohydrates by periodic acid shift. Then we have the steroid hormones. Steroids are derived from the cholesterol, and usually most of these are produced by the cortical part of suprarenal gland, comprising of the glucocorticoids, cortisol, aldosterone, mineralocorticoid, and gonadal hormone, which is dihydroepiandrosterone. Also, testosterone and estrogen, testosterone from the male, testis, and estrogen triol, or so droplets. So they are not membrane bound secretory vesicles. So we need to note as we describe these cells, we'll be able to. The other ones are amines, those are referred to as indole amines. The other amines are tyrosine, these are tyrosine derivatives, dopamine, epinephrine, norepinephrine. Epinephrine or epinephrine produced by the adrenal medulla, melatonin produced by the pineal gland, and T3, T4, this is thyroxine, which is produced by the thyroid gland. So these are the hormones that are amine. They are derived from tyrosine. And we'll be looking at the structure of this. So these are the nature of the hormones. So we need to understand this to be able to describe the cell and the content of the cell in terms of the organelles. Now we have specific hormones and cell organelles. Let's just look at this as a summary form. We have the amines. Those that produce the amines, these hormones here, are produced, the cells that produce this will be very rich in mitochondria and smooth endoplasmic reticulum. Mitochondria and smooth endoplasmic reticulum. Some of the final enzymatic activity for the production of the hormones occur within the mitochondria. Steroid hormones, the cells are normally very large, polygonal cells. They are eosinophilic staining, so they stain for acidic dyes. That's why it's eosinophilic, with very active chromatin within the nucleus. They are pale staining. Pale staining because during preparation, when you use H&E, usually the lipid within the cell is removed with the alcohol, and therefore those parts appear as pale staining. And because of that, the areas where the lipids are will appear as empty vacuoles. So the cytoplasm appears vacuolated. vacuolated. In these particular steroid-secreting cells, they have very well developed smooth endoplasmic reticulum. Very well developed smooth endoplasmic reticulum, and mitochondria that have a tubular crystal. You know the crystal of the mitochondria, they can be rod-shaped, but these are tubular in nature. So the mitochondria is where the final breakdown formation of the hormone actually takes place. Golgi apparatus is very poorly developed. So this is wrong spelling, poorly developed, because the hormone is not in a vesicle form. The hormone is in lipid droplets because it appears as lipid droplets. For the proteins, these are the same as the peptides secreting cells. They are very well developed rough endoplasmic reticulum. And because of that, they are basophilic in staining. They stain very deep basophilic. The rough endoplasmic reticulum is for synthesis of proteins. Ribosomes are attached to rough endoplasmic reticulum. We have the Golgi apparatus for the final packaging and also provision of the vesicles. And mitochondria, just like what we had said before, where we have final production of the provision of the energy. The peptides, the cells are usually polygonal or columnar shaped and are basophilic. Just like what we have in the amine form, they are basophilic. Basophilic because they have well-developed rough endoplasmic reticulum, the ribosomes. They also have Golgi apparatus, mitochondria, and microtubules. Remember, microtubules are very important in intracellular transport. Apart from this, we also have some common organelles. Some of the common organelles will include the Golgi for packaging, mitochondria, microtubules for intracellular transport, mitochondria to provide the motive force, the energy, and also we have the mitochondria, sometimes enzymes are located within the mitochondria. They are of different shapes. We have talked about polygonal cells. Some of them appear cuboidal, like in the thyroid gland. They are polygonal, like in the suprarenal gland. Most of the steroid producing are polygonal. They could be columnar, like in the parathyroid gland, or they can be stellate in shape, what we say in the pineal gland, where the cell has several processes that produce melatonin. So there will be different shapes, and these cells are attached together by junctional complexes. And each cell is exposed to a blood vessel, very close to a blood vessel, because the hormone is supposed to be transported by blood. Now, for embryological origin of these endocrine organs, they come from all the germ layers. In a summary, they all come from all the germ layers. I'll just mention each of those and which ones come from what, but overall, they come from all germ layers. The hypothalamus is neuroendocrine, is from neuroectoderm, from the diencephalon. The pineal gland is a neural ectoderm. Remember, the ectoderm eventually divides into surface ectoderm and neural ectoderm. The neural ectoderm developed to form the nerve tissue and neurocrest cells. The cells between the neural ectoderm and surface ectoderm will be referred to as neurocrest cells. And then the rest of the cells will form the epidermis. The hypophysis comes from two sources. There's a neural ectoderm, the neurohypophysis from the diencephalon, and then the glandular part, the adenohypophysis, is from the oral ectoderm. And this is from the rat case pouch. evagination of the oral ectoderm, forming Rathke's pouch. And then this Rathke's pouch migrate towards the neural ectoderm and fuse together now to form the gland that we refer to as the hypophysis. The neural ectoderm retains its conduct to the brain. The adenohypophysis does not retain its conduct with the oral cavity. The thyroid gland is from the third pharyngeal pouch, so it is endodermal in origin. The suprarenal gland, the cortex, is from the mesoderm, splanchnic mesoderm. The medulla is from the neural ectoderm as migration of the neurocrest cells. So medulla is of neurocrest origin. The pancreas is from the foreguard endoderm as evagination of the endoderm at the distal part of the duodenum. The parathyroids, we have two parathyroids. We have the superior parathyroids from the fourth pharyngeal pouch, the inferior parathyroids from the third pharyngeal pouch. The gonads are from the intermediate mesoderm, formed from the gonadal ridge. Diffuse neuroendocrine system, most of the cells secrete peptides and there are neurocrest derivatives. So they originate from the neural ectoderm. So you can see that we have cells from the ectoderm, we have cells from the mesoderm, and we have cells from the endoderm. Let's now look at the hypothalamus. The hypothalamus, like we have said, is a part of the brain below the thalamus, and the roof of the hypothalamus is formed by the third ventricle. So that is the roof of the hypothalamus. So the hypothalamus actually not the roof, but is surrounded. The hypothalamus surrounds the third ventricle. The hypothalamus, for the endocrine purpose, will be looking at the hypothalamus again under the nervous system. But for endocrine purposes, the hypothalamus have several aggregates. It's nerve tissue, but these are specialized secretory neurons. They are secretory neurons. They are not myelinated. Because they don't transmit an impulse. The cell bodies of these neurons aggregate to form some nuclei. Aggregates of cell bodies in central nervous system, we call the nuclei. So we have two types of nuclei. We have those with very large cell bodies that we call magnocellular nuclei. These are the two. We have paraventricular adjacent to the ventricle. These ones have very large cell bodies. And then supraoptic nucleus above the optic chiasma. These secretory neurons secrete two hormones, oxytocin and vasopressin. The hormone is then transported from the hypothalamus, from this nuclei, to the neurohypophysis. So the axons extend all the way from the hypothalamus to the neurohypophysis, where the hormone is released, stored and released. The hormone is in a form of, within the cell, we have visible structures that we call the herring bodies. Herring bodies contain the hormone, and the protein called neurophysin is very important in that transport of the hormone to the neurohypophysis. So this is by exoplasmic flow. We'll be looking at the neurohypophysis. The other cells, the other nuclei, have small cell bodies. Small cell bodies. So we call them parvocellular. Parvocellular. This includes ventromedial nucleus, dorsomedial nucleus. We have the infundibular nucleus, the tubal nucleus. These nuclei have small cell bodies. Again, those neurons are secretory. They secrete releasing hormones. These releasing hormones are transported by very short axons to the median eminence, where we have the primary capillary plexus. So they are released at the primary capillary plexus. The primary capillary plexus pick up those hormones, also called releasing hormones or inhibiting hormones. The releasing hormones are also referred to as liberins. The inhibiting hormones are referred to as statins. So the primary capillary plexus picks it up, transports it to the adenohypophysis through the hypophyseal portal system. So we have portal veins that transport the hormone to the adenohypophysis. The hormones then leave the blood vessels and stimulate the cells within the adenohypophysis. And those cells now produce hormones which are transported to the target organs. So this is what we have as the hypothalamus. Magnocellular and parvocellular nuclei. So we have talked about the hypophysis, and the hypophysis has two parts. The adenohypophysis from the Rathke's pouch. You can have three parts of the adenohypophysis. We have a pars tuberalis that come to surround part of the infundibulum, the neural part. Then we have pars intermedia between the neurohypophysis and the adenohypophysis, where both neural tissue and glandular tissue are mixed together. And then we have pars distalis, which is the largest part of the parts. We have the median eminence. In the median eminence, this is where the capillaries lack a blood-brain barrier. Blood-brain barrier. So the hormones can get in very easily into the blood at the median eminence. And then the infundibulum, which is the stalk that connects the distal part of the neurohypophysis to the hypothalamus. And then pars nervosa. The pars nervosa is the large part where we have the storage of the oxytocin and vasopressin, and also release at that part. So this is to illustrate in a nutshell the connection between the hypothalamus and the hypophysis. For the neurohypophysis, we have the hormones, supraoptic nucleus and paraventricular nucleus, which are large cell bodies, magnocellular, release the hormone oxytocin and vasopressin. It is transported through this hypophyseal tract to the pars nervosa, where the hormone is released into the blood. Then we have the parvocellular. These are small cell bodies. They pick the hormone. The hormone is synthesized in the cell body. It is transported and released at the median eminence to the primary capillary plexus. And then from the primary capillary plexus, those releasing or inhibiting hormones are then transported to the adenohypophysis by the hypothalamo-hypophyseal portal system. And then at the secondary capillary plexus, the hormone is released and stimulates the cells within the hypophysis now to produce hormones. Among the hormones produced are prolactin, that is very important in development of the mammals, that are very important in the growth of bones, muscles, and adipose tissue in the body. We have luteinizing hormone, very important in the release ovulation, and also very important in secretion of testosterone in the testes. We have follicle stimulating hormone, very important in the development of the follicles in the ovary, and also very important in the functioning of the Sertoli cells within the testes for the production of spermatozoa. Thyroid stimulating hormone to the thyroid gland that now secrete the hormone thyroxine. Adrenocorticotropic hormone that stimulates adrenal cortex. And then oxytocin and ADH, which we talked about. Oxytocin to the uterus for contraction of the uterus, and then stimulation of the epithelial cells within the mammary gland for milk let down. Apart from this, it also supplies the testes, the smooth muscles within the testes for the transportation of spermatozoa. And then antidiuretic hormone in the kidney that is very important in reabsorption of water for the concentration of urine. So this is a very important connection between the hypothalamus and the hypophysis. Hypophyseal axis, but if it goes to the uterus, ovaries and testes, then we refer to it as hypothalamo-hypophyseal gonadal axis. Hypothalamo-hypophyseal gonadal axis. This is the hypothalamus. This is the infundibulum that connects it. This is now the pars nervosa and pars distalis of the hypophysis, sorry. Now, for the development of hypophysis, we are from the diencephalon and also from the roof of this oral cavity at Raschke's pouch. So this continue on. Eventually, this detach from the roof as Raschke's pouch and come to lie close to where we have neurohypophysis, and where the two cells are very closely together, the cells mix up. So the adenohypophysis now comes to form the pars tuberalis, pars intermedia, and pars distalis. Note connection, continuous connection by the infundibular stalk to the hypothalamus. Note also the formation of extension that comes to end of this as pars tuberalis, pars intermedia, and pars distalis. In the pars intermedia, we have some colloid remnants. For the hypophysis, also called pituitary gland, we have This is the neurohypophysis. This is the pars intermedia. This pars distalis, and then the pars tuberalis. This is the infundibulum. For the cell structure, of course, the hypophysis is covered by a connective tissue capsule. And usually you can group the cells into those that take up stains that we refer to as chromophils, and those that do not take up stain that we refer to as chromophobes. Among the chromophils, we have the acidophils that have affinity for acidic dyes, and basophils that have affinity for basic dyes. Among the acidophils, these cells we call somatotropes that produce growth hormone under the stimulation of somatotropin-releasing hormone from the hypothalamus. We have the mammotropes, that's very important in mammary gland, that we have mammotropin-releasing hormone from the hypothalamus stimulating the mammotrope cells. These are acidophils. And then for the basophils, we have gonadotropes that are stimulated by gonadotropin-releasing hormone from the hypothalamus. The thyrotropes stimulated by thyrotropin-releasing hormone from the hypothalamus. These ones are from the parvocellular nuclei of the hypothalamus. Parvocellular nuclei of the hypothalamus. We have the corticotropes that produce corticotropin-releasing hormone from the hypothalamus. These are releasing hormones, also referred to as liberins. At the same time, when we have the negative feedback mechanism, cells within the hypothalamus will release inhibiting hormones. So we will have somatotropin releasing hormone, inhibiting hormone, mammotropin inhibiting hormone, gonadotropin releasing inhibiting hormone. Sometimes we call those statins. So those will inhibit secretions from the hypophysis. What happens when you have a tumor of the hypophysis? When the glands of the hypophysis have tumor and they grow, they have cancerous tissue, they grow, they produce excess hormones. Specifically, two cells that are of importance here. We have those that produce the vasopressin. Vasopressin, that is the paraventricular and also supraoptic nuclei. Those ones might produce excess vasopressin. So because of excess vasopressin, we'll have a lot of water. Diuresis. We'll have diabetes insipidus. So if they are damaged, so they'll have diabetes insipidus, where you have frequent passage of urine. And then growth hormone deficiency. If there's damage to the growth hormone cells, then you have dwarfism. People are very short stature, dwarfs. And then if we have excess growth hormone, before 20 years, usually the hypophyseal plate closes by 20 years. So if the hormone occurs before 20 years, then the hypophyseal plate continues to grow. The bones become very long. Then you end up with gigantism. Gigantism, very tall people, 8 feet tall, very tall people. But if after the epiphyseal plate has closed and we have excess production of the hormone, then we have acromegaly. The bones become very large, very thick. We have the bone, the face becomes very thick. The skin becomes very thick when you have those excesses production. So these are some of the disorders that can occur associated with the hypophysis. This is the hypophysis. We can note here the 3 cell types. We have these cells, these are basophilic cells. Note that the cells are arranged in a cord, they are in cord, And in between these coats, we have sinusoidal capillaries with some little connective tissue supporting the blood vessels. The orange staining cells are acidophils. The orange staining cells are acidophils. And then these other cells which do not take up the stain are the chromophobes. Chromophobes form 50% of the population. So we can notice here the acidophils and basophils. Acidophils and basophils. This has been stained with orange G. In between that, we have capillaries that are sinusoidal. Here, notice the cells are arranged in a cluster. So cells are arranged either in a cord or a cluster. In this part, notice the acidophils, and all these are chromophobes. Chromophobes. They do not, the cytoplasm is not staining with acidic or basic dyes. And these are the basophils. Here, notice the cells are arranged in a cluster. In a cluster. And in between this, we have the capillaries. Just to show the chromophobes with H and E, notice the acidophils deep staining, the chromophobes not picking up any stain, and basophils basic dyes because it contains a lot of rough endoplasmic reticulum. This is the pituitary gland, the neurohypophysis. This is where oxytocin is stored and released, and oxytocin is stored in the form of herring bodies. This we refer to as herring bodies, H-E-R-R-I-N-G. Apart from the termination of the neurons from the hypothalamus, from the supraoptic and the paraventricular nucleus, we also have pituicytes. Pituicytes. Pituicytes are glial cells. Pituicytes are glial cells because this is nerve tissue. Neurohypophysis is an extension of the diencephalon, so it is nerve tissue, and therefore we have some pituicytes, which are glial cells. So that is about the hypophysis and hypothalamus. The pineal gland, we said, is P-shaped. That's why it's called pineal. It's in the roof of the third ventricle, connected by a pineal stalk to the brain. We have a pineal stalk to the brain, and we have pineal recess, a small depression, and the commission, posterior commission, and also pineal recess. That's where it's located. For the structure, we have a dense connective tissue capsule, just like what we have talked about. But this tissue, because it's attached to the brain, continues on with the leptomeninges, that is the dura and the pia mater. The capsule penetrates by form of septa into the organ, giving it a supportive framework. The parenchyma usually contain pituicytes, pinealocytes, and astrocytes. Pinealocytes and astrocytes. Now, the parenchyma has a lot of blood vessels because the blood is supposed to transport the hormone that is released to the perivascular space. In old age, after puberty, the pineal gland starts to have congregations, what we refer to as the pineal sands, or corpora amarcea, the pineal gland, pineal sand. The pinealocytes are pale staining, and they have a lot of processes. We talked about them having stellate in shape with a lot of processes. And these processes they penetrate into some what you call canaliculi. These are very narrow ducts. Very narrow ducts. And processes project into these narrow ducts. These narrow ducts are thought to be channels for the transport of the hormone, melatonin. The predominant organelles that we have here, what they secrete is indole amines. Indole amines. So we have mitochondria, rough endoplasmic reticulum as predominant, and then Golgi apparatus. We also have smooth endoplasmic reticulum, centriole, and lysosomes. But the predominant one here is rough endoplasmic reticulum and mitochondria. Apart from this, we have, of course, microtubules for the intracellular transport. The secretory granules contain melatonin, and this melatonin is produced by neural stimulation. This is from the suprachiasmatic nucleus, which is usually referred as the biological clock. So during the darkness, the suprachiasmatic nucleus melatonin is produced in the darkness. So this is what we are talking about in terms of very close to the capillaries, and then we have the processes. This is just a section which we'll be looking at these slides specifically. Look at the blood vessels. Then we have the pinealocytes come very close to the blood vessels. Some of their processes come close to the blood vessels. The others are the new glial cells. This is the stalk that connects the pineal gland to the brain, the habenular commissure. And then we have in the adult and also after puberty, we have pineal sand. So when you look at the pineal gland, we should be able to identify the pineal sand. What will be the function of melatonin? We have said melatonin, usually when you have light, when there's increased intensity of light in the retina, from the retina, some of the neurons end up at the suprachiasmatic nucleus, which we have said is the biological clock. So from the suprachiasmatic nucleus, some neurons end in the paraventricular nucleus in the brain. These ones usually are the autonomic nervous system. Autonomic nervous system. From here, the neurons go to the spinal cord, and then the superior cervical ganglia, that is sympathetic nervous system. And from this superior cervical ganglia, the neurons then stimulate the pineal gland to reduce the amount of melatonin. So when you have an increase in light, there's reduction in melatonin. So melatonin is produced during darkness. What will be the functions of melatonin? Mainly to regulate sleep-wake cycle. Sleep-wake cycle. Occasionally, when you have traveling over a long distance, many hours, and you're traveling east, you will have a jet lag. That jet lag is because of problems of melatonin. So to be able to now regulate your body, you take melatonin tablets because you have not slept for a long flight. It's also very important in inhibiting gonadotropin releasing hormone. Gonadotropin releasing hormone is usually a liberine that stimulates the hypophysis so that you can have release of luteinizing hormone and follicle stimulating hormone. So if we have very low melatonin levels, then that inhibition is stopped. So we have more gonadotropin releasing hormone. We have precocious puberty. A female comes to puberty very early, at a very early age. If we have very high levels of melatonin, then we have delayed puberty because there is a lot of inhibition of gonadotropin releasing hormone, and therefore inhibition of the release of luteinizing hormone and follicle stimulating hormones. So the follicle stimulating hormones, which are supposed to stimulate the ovary to start the cycle, now will be reduced, and that's why you have delayed puberty. So this normally is very important, not very important in man, but very important also in animals because of seasonal breeding. Seasonal breeding. In the males, it also regulates epididymal function. Epididymal function, which is very important in the maturation of spermatozoa. Maturation of the spermatozoa. So it will inhibit release of testosterone and therefore again delayed puberty if we have very high levels. If we have very low levels, then again what will happen is somebody comes to puberty very early because testosterone will be produced at an early stage. So these are the central endocrine organs. Our class was supposed to be 1 hour. I'll now invite any questions. We'll have to reschedule overviews for the remaining parts. Any questions? You can now ask your questions. Put it in the chat. Class reps, are you supposed to be going to another class now? Nothing?
No.
Dental?
No.
Physiotherapy?
We have a class at 11.
Okay, so we can continue to 11 instead. Consensus? We agree on to continue to 11?
Yes.
Thank you. So this is where deal with the central, and you can see how they influence the peripheral endocrine organs. How they influence peripheral endocrine organs. Okay, before we leave that, let me just make comments on this. Somebody wants us to talk about parvocellular. Now, parvo means small. Parvo means small. So the nuclei, the cell bodies of some secretory neurons, they do not transmit an impulse, but they secrete hormones, are small in size, and therefore they are classified as parvocellular neurons. And these ones here are within the dorsomedialis, ventromedialis, the infundibular, and the tubal nucleus of the hypothalamus. The parvocellular neurons have very short axons. The axons terminate in the primary capillary plexus, where they release the releasing hormones, which we talked about. For every hormone released by the pituitary, there is a releasing hormone which stimulates its production. So that is released at the median. The primary capillary plexus is at the median eminence. That's why I said at the area of the median eminence, the blood vessels, the capillaries there do not have a blood-brain barrier. Once the hormone has been released to the primary capillary plexus, it is transported to a secondary capillary plexus in the adenohypophysis. Any blood vessel that joins two capillary plexus is what we call a portal system. This is a portal system joining the hypothalamus and the hypophysis. That's why we call it hypothalamo-hypophyseal portal system. At the secondary capillary plexus, the releasing hormones are now released to the cells. And those cells are the acidophils and the basophils. The specific target for each of those cells depends on the releasing hormone, because they have receptors for those releasing hormones. So if it is a follicle-stimulating hormone, gonadotropin hormone will only stimulate those cells that will produce follicle-stimulating hormone and luteinizing hormone. That is the gonadotropes. If it is a growth hormone, the growth hormone releasing hormone will only act on the growth hormone cells, the somatotropes. So that's what you're talking about in terms of parvocellular. Magno are the large cell bodies that will produce oxytocin and vasopressin. So I've talked about that, and I've talked about pars tuberalis, intermedia, and distalis. Now, for pars tuberalis, intermedia, and distalis, the Rathke's pouch, when it comes in contact with the diverticular from the diencephalon, the floor of the diencephalon, a part of it comes to envelope the infundibulum. That is the stalk. And that is what we call pars tuberalis. Pars tuberalis. And then at the part where the neural part and the glandular part are meeting, usually the glandular tissue mix up with the neural part. And that's what we're calling it pars intermedia. And then the rest of purely glandular tissue is what we call pars distalis. Fine, if there are any questions, again, we'll continue looking at them. So we've seen the effect of hypothalamus, the hypophysis, and now regulation by the regulation of the peripheral endocrine organs. One of the peripheral endocrine organs is the thyroid organ. The thyroid organ originates from the floor of the third pharyngeal pouch. Floor of the third pharyngeal pouch. This is normally marked in the tongue by a depression at forming the anterior 2/3 of the tongue and the posterior 2/3 of the 1/3 of the tongue, that we refer to as foramen cecum. So the endodermal cells grow downwards, they vaginate downwards and come to lie in the neck region. In the neck region over the thyroid cartilage. At the distal part, they form two lobes, they divide to form two lobes, joined by a very small area that we refer to as the isthmus. The connection to the tongue usually disappears. Occasionally, however, we might have some remnants of the thyroid gland in the tongue. Sometimes we call thyroglossal cysts or thyroglossal duct that continue on to the thyroid gland. Can you stop talking, please? Mute your microphone. So the thyroid gland is covered by a connective tissue capsule. It's covered by a connective tissue capsule, just like the other organ. So it's located over C5T1, that's for gross anatomy, covered by infrahyoid muscles, supplied by the superior thyroid artery, inferior thyroid artery, and the isthmus is covered by thyroid ema from thyrocervical trunk. Can you please stop, mute your microphone, whoever is not muted? For the structure, we have a capsule, we have the stroma, which is connective tissue, just like what we have talked, supporting blood vessels, nerves, and lymphatics. The parenchyma consists of thyroid follicles. So the functional unit of the thyroid is a follicle. Between those follicles, we call that parafollicular area, which contain parafollicular cells. The follicles are lined by an epithelia that vary in shape. It can be simple cuboidal to low columnar, depending on the functional status of the thyroid. We have a very well-developed capillary network around the follicles because the hormone is to be transported by blood. The cells, which are the thyrocyte, I've said are cuboidal or columnar in shape, and their main function is synthesis and also storage of T3, T4. The cells synthesize the hormone. The hormone is stored in the form of the colloid, as a thyroglobulin. That's the colloid. Between the follicles and also sometimes embedded between the follicular cells, we have C cells, what we call C cells, and parafollicular cells. Parafollicular cells secrete a hormone called calcitonin. Calcitonin lowers the calcium levels in blood. It lowers calcium levels in blood by promoting deposition of calcium in the bone, by promoting reabsorption, not reabsorption, secretion of calcium in the kidney, and by inhibiting reabsorption of calcium in the digestive tract. So in that way, you are able to lower the calcium levels in blood. This is a thyroid gland. This large connective tissue area is what we call the septa, and the septa contain blood vessels. The septa then penetrate into the organ, dividing it into lobes and lobules. So each of these is a lobule. In each of the lobules, we have a follicle. So this is the functional unit of the thyroid gland. And the follicle contains colloid, thyroglobulin, colloid, and is lined by simple cuboidal to low columnar, depending on the functional status. In an inactive thyroid gland, there is an increase in the amount of colloid because the hormone is stored at thyroglobulin in an active gland. In an hyperactive gland, the colloid is then taken in by endocytosis. The colloid is taken in by endocytosis, and the hormone T3, T4 is released into the blood, and there's recirculation of the iodine and also the globulin part. So that way, the colloid is removed from the center. The cells, therefore, become almost columnar. Like I've said, low columnar. So this is a hyperactive thyroid gland. The other one is inactive thyroid gland. Around the follicle, this is a follicle. Around the follicle, we have blood vessels, a very well-developed capillary network comprising of fenestrated capillaries. Between the follicles, we have parafollicular cells that secrete calcitonin. So this is an inactive thyroid. Notice the cells are cuboidal in shape, a very large colloid containing thyroglobulin. Notice again cuboidal in shape, connective tissue surrounding the follicle, which is a functional unit, and this connective tissue contains a very well-developed capillary network that surrounds the follicle. And in between those follicles, we have parafollicular cells. Again, notice an inactive follicle, but just to demonstrate the capillary network, what appears as red here, these are capillaries that form a very well-developed network around the follicle, specifically for the transport of the hormone, and also transport of the calcitonin produced by the parafollicular cells. These are parafollicular cells. At high magnification, note the capillaries. Note a high content of colloid because it's inactive. Now, this has been stained to demonstrate calcitonin. So what appears as brown, those are cells that contain calcitonin. Those will be the parafollicular cells. They can either be between the follicles and also embedded between the follicular cells. So those we refer to as C cells. C cells secreting calcitonin. What can go wrong? Sometimes we have a remnant of the connection, some remnant within the tongue or within the neck, upper neck region, of part of the verticula that was eventually to form the thyroid gland. So we have thyroglossal cysts, which can be in the tongue. But we can also have iodine deficiency. How is thyroxine formed? Thyroxine, first we have the protein synthesis, the globulin. Globulin is synthesized by rough endoplasmic reticulum. It is transported to the Golgi apparatus. It's packaged in the Golgi apparatus with some carbohydrate moieties. Then that is at the apical part of the cell, we have iodination of tyrosine moieties, iodination of the tyrosine. But before that, we have the iodide uptake. The iodide uptake by the cell. The iodide is then converted to iodine, mainly at the apical. Sometimes they say at the apical part of the cell. Some people say it is within the lumen. Some literature is not completely clear. The iodine, then we have iodination of the tyrosine moiety by thyroid peroxidase enzyme. So that forms what we refer to as monoiodotyrosine. Monoiodotyrosine. And also diiodotyrosine. So after this iodination with 3 or 1 iodine, we then have coupling. And coupling is where we have a combination of monoiodothyronine and diiodothyronine forming triiodothyronine, T3. Or you have diiodothyronine having 2 iodine. The 2 of them can combine to form T4, tetraiodothyronine. So these are the enzymes which are stored within the colloid as thyroglobulin. During the release of the enzyme is taken up. Again, the coupling and also the formation of the monoiodothyronine and diiodothyronine is occurred by thyroid peroxidase enzyme. During the uptake of the enzyme, the colloid is taken up by endocytosis. That is endocytosis. It combines with a lysosome. So lysosomes are very well developed. Within the lysosome, we have proteases. The enzyme proteases that breaks down the protein tyrosine so that the T3 and T4 are released. T3 and T4 are released. And then the globulin part, the protein part now is taken up to the liver. Sometimes we also have within the liver, the enzyme can be broken down to form monoiodothyronine and diiodothyronine within the liver or the kidney. T3 is the most active part, and that is very important for metabolism of the body. Thyroxine is very important in metabolism. So when you have iodine deficiency, the gland will try to grow, the cells will try to grow bigger, become bigger, to try to produce as much hormone as possible. Because of that, there is enlargement of the thyroid gland that we normally see as goiter. We can have hypothyroidism, too much production of the hormone T3, T4, what you call Graves' disease. And in hypothyroidism, there's a high heart, there's hypertension, and we also have rapid palpitation of the heart, rapid beating of the heart, and intolerance to temperature. Or we can have less production of the thyroid hormone, hypothyroidism. So in hypothyroidism, remember we have very low thyroid hormone in circulation. Sorry, there is no R. Hashimoto's, no R in Hashimoto's. So we have the condition which is called myxoedema. And usually, because there is metabolic rate is very low, the person is intolerant to temperature, cannot withstand temperature because it's very low, body is very low temperatures. And the person is inactive also. The other gland that is very closely associated with the thyroid is parathyroid. Parathyroids are two, we have said, superior and inferior parathyroid. The superior parathyroids originates from the fourth pharyngeal pouch, so it is endodermal. It is dragged along by the superior parathyroid goes on to the thyroid gland. As the thyroid gland also grows, it moves on together with the thyroid gland and ends up to lie very close to the thyroid. The inferior parathyroids originate from the third pharyngeal pouch, close to where the thymus originates. So during the migration of the thymus, it tugs along the thymus and eventually comes to lie inferior within the thyroid gland, the capsule of the thyroid gland. So that is the origin of the two glands. The parathyroid can appear individually, normally 4 parathyroids. The parathyroids can appear individually, covered by a capsule, or they are embedded within the capsule of the thyroid gland. It comprises of 3 cell types. The principal cell type, also called chief cells, are the cells that produce the hormone, parathyroid hormone. They are epithelial arrangement, usually columnar or polygonal in shape. Columnar or polygonal in shape. We have free ribosomes, large Golgi apparatus, secretory granules, some lipid droplets, and some glycogen. That is when it is active, when it's producing the hormone, parathyroid hormone. So those are chief cells, slightly pale staining because of lipids, basophilic because of the ribosomes. The other cell types are clear cells. And clear cells are thought to be a form of chief cells that have excess glycogen. This can be demonstrated with the PAS staining, periodic acid shift staining. They are also thought to be degranulated cells that have shed off the hormone, parathyroid hormone. The other cell types are oxyphils. The oxyphil cells, they are polygonal, they are large, small nucleus, some mitochondria, glycogen granules are also present. Free ribosomes are in the cytoplasm. And these appear at puberty and increase with age. Because of that, they are thought to be degranulated cells, but they're also called oncogenes. Oxyphils, the cytoplasm is full of mitochondria, and therefore the oxyphil are deeper staining, basophilic in nature. The parathyroid gland also contain adipocytes, white fat, embedded amongst these other cells. What is the functions of the parathyroid? One, production of parathyroid hormone. And the parathyroid hormone increases the calcium levels in blood and therefore maintains calcium levels in blood. It increases calcium levels in blood in three ways. One, it can promote osteoclastic activity, osteoclast. So there is a resorption of bone, calcium from the bone by osteoclasts. Two, it increases reabsorption of calcium in the kidney. Three, it increases absorption of calcium in the digestive tract. So overall, it increases the calcium levels in blood. So what happens when we have excess parathyroid production? If you have a tumor of the parathyroid gland, it means there is excess calcium. There'll be too much deposition within the bones. Well, too much reabsorption from the bones if you have parathyroidism. Too much calcium. The bones will become very weak. So there's pain in the bones. There'll be deposition of Sometimes calcium, because there is too much of it in the kidney, so you can end up with kidney stones. What about when you have hypoparathyroidism? Sometimes when they remove the thyroid gland, they might remove parathyroid accidentally. Then there is very low calcium levels in blood, and therefore you end up with muscle cramps because calcium is very important for contraction of the muscles. Apart from muscle cramps, you will also end up with the tetanic contractions, tetanic contractions of the muscles. So that's what we have as some of the disorders of the parathyroid gland in case either there is less production or a tumor stimulating overproduction. The thyroid gland is not regulated by the hypophysis or hypothalamus. The functioning of the thyroid gland is regulated by the amount of calcium in blood. If there is high calcium in blood, then calcitonin takes in to reduce the calcium. If there's low calcium in blood, parathyroid then increase calcium levels in blood in the three ways we have talked about here. This is the thyroid gland on the right. This parathyroid gland, like I said, they're very closely associated. They have their own separate capsule. Here we have two cell types. You look at the cytoplasm, the cells with the deeper staining cytoplasm. These ones here are the oxyphils. Oxyphils have deeper staining cytoplasm because of a lot of mitochondria. The cells that are light stained cytoplasm, very light stained, these ones are chief cells. Chief cells and also clear cells. Chief cells and clear cells. And notice the blood vessels. Notice the arrangement of the cells. They are epithelioid in nature. The pancreas. We have done the pancreas under the digestive system. And we said we have two parts of the pancreas, the exocrine pancreas and the endocrine pancreas. Exocrine pancreas, the acini secretes the enzymes to the duct, to the intercalated duct, and then striated duct. The enzyme is transported and emptied into the digestive tract for the digestion of lipids, digestion of carbohydrates, digestion of proteins. Scattered among the endocrine parts of the pancreas, we have the isolates of Langerhans. Isolates of Langerhans. The isolates of Langerhans are surrounded by a capsule, a thin connective tissue capsule. It's a highly vascularized region. They are pale staining compared to the other cells of the exocrine pancreas. And we have several cell types. In histology, normal hematoxylin and eosin staining, we are not able to distinguish these cell types. You have to do some immunostaining to be able to distinguish them. We have the A cells, alpha cells, comprising 20%. This secrete the enzyme glucagon. And glucagon is very important in breakdown of glycogen in the liver to form glucose. The majority of the cells are the B cells or beta cells, constituting 75%. And this secrete the enzyme insulin. And insulin is very important in glucose metabolism. Insulin promotes uptake of glucose by the cells and utilization of glucose by the cells. Where we have less insulin in circulation because of damage to B cells, then you end up with diabetes mellitus. Diabetes mellitus. Either type 1 or type 2 diabetes mellitus. This can also occur where we have insensitivity of the cells to insulin. Adequate insulin is produced, but the receptors within the cells are missing. Then it behaves like you have less insulin in the body. The other cell types are the D cells or delta cells, comprising about 5%, and this secrete the hormone somatostatin. Somatostatin inhibits both A cell and B cell. It inhibits both A cell and B cell. What is glucose used for? Production of energy and growth. The other cell types are the P cells, and P cells produce pancreatic peptides. Pancreatic peptides. These ones are also in the acinar cells. Pancreatic peptides. And they also produce ghrelin. The ghrelin is the hormone that makes you feel hungry. So if your stomach is empty, there's more production of ghrelin, and therefore you feel that you are hungry and you must eat. Also, the amount of ghrelin circulating in blood will stimulate the satiety center, the feeding center in the brain. Of these cells, B cells are the majority, and they occupy the central part of the isolates of Langerhans. The A cells, the D cells, and the P cells are the minority. So this one, after immunostaining in man, this is how they have come about to map out the various cell types. The blue cells are insulin secreting cells, so they are B cells. These other cells are the glucagon secreting cells. Notice they are normally at the peripheral part, but the majority of the B cells are central. Pancreatic peptide cells also are at the peripheral, secreting ghrelin. And then somatostatin, also the D cells. You can notice there are very few D cells scattered amongst the B cells. After immunostaining, this is what we see. This is immunostained for A cells, alpha cells, and you can see they are peripheral. Notice the blood vessels here and also blood vessels here. So the cells are arranged in clusters and coats with blood vessels around them. This one has been stained to demonstrate insulin using anti-insulin, insulin as a marker. And we see that most of the cells are centrally placed and they form the majority of the isolates of Langerhans. In this slide, this is the isolate of Langerhans, very well vascularized. We have a thin connective tissue capsule separating it from the acinar cells. These are the acinar cells of the exocrine pancreas. And this is the isolates of Langerhans. From this, you can see different staining characteristics. Some of them The cytoplasm doesn't seem to stain much. In some, the cytoplasm is staining very deeply. So this just to demonstrate with H and E that we have several types of cells within the isolates of Langerhans. Using a different stain, again, the majority here are the B cells and then a few of the A cells. But of importance is the arrangement of the cells and also the blood vessels. This is another slide. You'll be looking at this slide. This is the isolate. Notice the connective tissue capsule, thin connective tissue capsule. The blood vessels, what appears as yellow, these are blood vessels, the red blood cells within capillaries. And then the arrangement of the cells are clusters and cones. Using a different stain again to demonstrate, the isolates of Langerhans are scattered amongst endocrine, exocrine cells. Most of the isolates of Langerhans are in the tail of the pancreas. Notice the arrangement here, the blood vessels. These are blood vessels, and you can see the cells are very close to the blood vessel because the hormone must be transported by blood vessels. So they appear as large and small clusters of cells. Notice these are very small cluster of island of cells. These are larger cluster, and so on. Again, the same isolate, a pale staining region scattered among exocrine. So that is the pancreas, isolates of Langerhans. How about suprarenal? The suprarenal glands, it used to be called adrenal, but it was found that it is actually above the kidney, at the superior pole of the kidney. So it was referred to now as suprarenal gland. Suprarenal gland is of dual origin. The cortex originates from the splanchnic mesoderm. The medulla originates from the neurocrest cells. So it is neural ectoderm. We have what we call the fetal cortex, which developed very early as epithelial cells. They form something like epithelial cells. So this fetal cortex will be surrounded by a cluster of other cells. The fetal cortex after birth undergoes involution. So the cells disappear. And this thin cluster of cells surrounding the fetal cortex then differentiates to form all the three layers of the cortex. Therefore, that we refer to as the definitive cortex. Definitive cortex. So by the third year, we are able to identify all the three layers of the cortex. Which are these layers of the cortex? And how are they arranged? The layers of the cortex are named according to the arrangement of the cells. The outermost layer, which is called zona glomerulosa, the cells are arranged in clusters, nests, or clumps. Clusters, nests, or clumps. They are polygonal in shape, centrally placed nucleus with an active chromatin nucleus. They secrete mineralocorticoids. Aldosterone. And this is under the influence of renin-angiotensin system. So the zona glomerulosa is not influenced by adrenocorticotropic hormone from the hypophysis. It is influenced by the sodium blood concentration. Aldosterone promotes reabsorption of sodium in the distal convoluted tubules of the kidney. And you then have passive reabsorption of water, reabsorption of sodium, and passive reabsorption of water. The next layer, but we need to note also that the zona glomerulosa, if we have the pituitary gland removed, it does not influence the cells in the zona glomerulosa. The thickness of zona glomerulosa does not change. If we have, however, a sodium deficient diet, if somebody takes a sodium deficient diet for some time, then zona glomerulosa cells undergo hypertrophy, they enlarge. So the thickness of zona glomerulosa increases because it is trying to produce a lot of hormone to be able to reabsorb sodium. So if we have a diet with excess sodium production, then zona glomerulosa thins out. And therefore, it is concluded that zona glomerulosa is not influenced by adrenocorticotropic hormone. The next layer is zona fasciculata. In zona fasciculata, the cells are arranged in fascicles. They are codes. They are arranged in fascicles. They are codes. They are polygonal. And just like any steroid secreting cell, the cells appear vacuolated. They are pale staining. Refer back to what we discussed earlier. Zona fasciculata cells secrete glucocorticoids. Glucocorticoids are anti-inflammatory and also promote carbohydrate metabolism. Cortisol is an example, and also cortisone. In zona reticularis, the cells are arranged in anastomosing codes. And in this zone, the cells stain deeper because they contain lipofuscin granules. Zona reticularis cells secrete the hormone they call the gonadal hormone, dihydroepiandrosterone hormone. So these are the hormones and these are the cells. So this is a global view of the suprarenal gland covered by a thick connective tissue capsule. Sometimes within the capsule, we have the sympathetic and parasympathetic ganglia. We have ganglia, parasympathetic ganglia, sometimes embedded in the capsule or very close to the capsule. So here we can see the two areas. This is the cortex and this is the medulla, cortex and medulla. And in the cortex, you can see the cells, different arrangement of the cells. The medulla here has very large, these large cavities is because all the blood from the cortex drains to the medulla and is drained to the medulla. So we have two blood vessels. The blood vessels enter the capsule. They break down into capillaries, fenestrated capillaries. They pass between the cells and eventually end up in the veins in the medulla. Some blood cells bypass the cortex and go directly to the medulla and form a capillary network to supply the medulla with the nutrients. The medulla is, I've said, originate from the neurocrine cells. So they contain what we call chromaffin cells. They reduce potassium dichromate. That's why it's called chromaffin cells. They are sometimes also called agendaphin cells. Silver, they reduce silver. So columnar cells, which are chromaffin cells, very closely in contact with the blood. Amongst those, we also have sympathetic ganglia, which are, the cells are thought to be modified postganglionic neurons of the sympathetic nervous system from thoracic segment 11 to L1, the sympathetic nervous system. This is the global view. We have the capsule, we have the cortex, and we have the medulla. And in the cortex, we have cells arrayed in clusters. So this outer part here is the zona glomerulosa. Zona glomerulosa. And then we have this other area where the cells are arranged in fascicles in a line. This is now what we call zona reticularis. And then this part here is zona fasciculata. In zona fasciculata, we have less lipid in the cytoplasm and more lipofuscin granules. At high magnification, this is typical of the cells that secrete steroid. Centrally placed nucleus, the cytoplasm appears vacuolated. They are polygonal in shape. Polygonal, that means several, can be 5, hexagonal, pentagonal, but polygonal in shape, very large centrally placed nucleus. This is typical of all steroid secreting cells. This is glomerulosa. This is fasciculata. In between the fascicles, we have the sinusoidal capillaries. This one here is the medulla, and in the medulla, we have these cell bodies, which are the ganglionic cells. Ganglionic cells within the medulla. Sometimes what can happen, the suprarenal gland, you can have hypocortisolism, where you have cortisol as being produced, a lot of cortisol being produced. So in this case, you will have a lot of breakdown of proteins, so somebody becomes very thin because of that breakdown of proteins. We can have hyperaldosteronism, aldosterone. So when you have excess aldosterone being absorbed, then you have excess sodium being absorbed, excess water being absorbed, so the body becomes like edematous, or what you call anasaka. You have chronic adrenocortical insufficiency. That's insufficiency of all those hormones. You know the function of the hormone, and therefore, if we have insufficient hormones, it will have the opposite of what it is supposed to do. In the medulla, we have sometimes tumor of the medulla, of the chromaffin cells. So we refer to as pheochromatoscytoma. And if we have that, then we have excess production of epinephrine and norepinephrine. And remember, epinephrine and norepinephrine are stress hormones. So this is where you have some sympathetic, excessive sympathetic activity, restlessness, and so on. We have done the thymus, so I've just put it here to mention that thymus produce hormones, which are thymosin, thymolin, thymopoietin, and thymic neurofactor, very important in maturation of T cells. These are the thymic epithelial reticular cells, and we have covered that when we are dealing with the immune mechanism. For the endocrine ovary, we are going to deal with this again when you are dealing with the reproductive system, but just to mention that the primordial follicles usually are lined by flattened cells that we refer to as the follicular cell. This follicular cell, under the influence of follicle-stimulating hormone, will then differentiate from a cuboidal cell that we refer to as the granulosa cell. The granulosa cell secretes the hormone estrogen, under the influence of follicle-stimulating hormone. Now, the several granulosa cells develop, and we have the several layers, primary follicle, secondary follicle, and so on. But granulosa cells in general secrete estrogen and estradiol. These ones are steroid hormones. Immediately next to the basement membrane of these granulosa cells, we have the theca interna cells. Theca interna cells. And after ovulation, in which the oocyte, together with the corona radiata cells and cumulus operans cells are shed off, this lumen is now occupied by these granulosa cells and also the cells from the theca interna.